Hey guys! Today we're going to learn about viruses. So first of all, what is a virus? Viruses are one of the most tiniest organisms that you will ever come across. They're only about 20 nanometers long. Scientists classify viruses as non-living organisms because they lack the ability to reproduce and carry out metabolic pathways without a host cell. They were first discovered in the late 1800s by researchers that studied a plant disease called tobacco mosaic disease, in which the tobacco mosaic virus causes stunt growth and yellow spotted leaves in tobacco plants. Let's talk about the structure of a virus. A virus consists of a nucleic acid that can either be DNA or RNA, surrounded by a protein coat or shell known as a capsid. Some viruses also have a viral envelope made of glycoproteins and phospholipids that surround their capsid and helps them infect their hosts. Since viruses exist to wreak havoc on mankind, well, just kidding, maybe not really, but they are still responsible for detrimental diseases such as AIDS. Let's discuss how they are replicated. Viruses actually have a limited host range, so a cold virus can only infect your upper respiratory tract and not anywhere else. For now, we'll be focusing on the replication of bacteriophage, or bacteria infecting viruses. The phage will first attach to the receptor on a host or bacterial cell. When attached, the phage injects its own DNA into the host cell. The virus DNA uses the host's metabolic machinery to make copies of viral DNA and capsids. These viral components self-assemble within the host cell. Eventually, the bacterial cell is lysed or ruptured, and multiple copies of the virus is released. This is known as the lytic cycle, and phages that replicate only by the lytic cycle are known as virulent phages. Sometimes phages can incorporate their genetic material into the host DNA. Through this, when the host DNA replicates, the viral DNA, known as a prophage, also replicates. This is called the lysogenic cycle, and the host cell will not be killed as long as viral gene codes are repressed. So no new phages will be created, but certain conditions can cause the viral DNA to switch from the lysogenic to the lytic cycle and start copying viral components that results in the host cell's death. Bacteriophage that can replicate using both cycles are known as temperate phages. While we're talking about viruses, did you know that HIV is a retrovirus? Retroviruses are actually a special case. They're viruses that contain RNA, so they need to go through an extra step to incorporate their genome within a host cell. First, they use the enzyme reverse transcriptase to transcribe viral DNA from their RNA template. The new viral DNA incorporates with the chromosomes in the nucleus of a host cell. From there, the host cell treats the viral DNA as its own and becomes an HIV-producing factory.